So I just introduced this, the people that have contributed very much to the work. Most of the work that I'm going to present today has been performed by Mauro Sbroggiò in the lab together with Alessandro Bertero and Mara Brancaccio. Mauro is now in Madrid in the Ponte lab. And all of the more cardiological work has been done in collaboration with Giuseppe Lembo and Daniela Carnevale at the Neuromed Institute down in Pozzilli. So I was saying I'm coming to the sort of a very well-known problem in the cardiac remodeling, which is the ability of the heart to respond to several kinds of stress that increase the workload by adapting an hypertrophic phenotype in the muscle of the left ventricular wall. But if this stimuli of this increased workload persists for a long time, or it's overimposed with other kind of pathological situation, then what's happening is that this initial adaptive hypertrophy response evolved into a maladaptive hypertrophy with thinning of the muscle wall and so on. And I'm sure that I don't have to explain this to this particular audience, but this is just to put it in the general context of my talk. And one of the big questions, which is still very much open, I would say, is that of course, to identify the signaling which are responsible for these transitions and then eventually to this transition here. But what is surprising from the very large number of literature that is present in this field is that in many cases the signaling effectors that are involved in this event as well as in this event end up to very same kind of molecules. And there are a few very well-known and well-studied signaling pathway, like the ERK signaling pathway, the AKT signaling pathway, STAT3, as well as many others that have been implicated, both what is known in the adaptive hypertrophy as well as their involvement in the maladaptive hypertrophy. So the question is open, and I would like to discuss with you our small contribution in that particular area, coming with this relatively, not so well studied protein, because we discovered it years ago, this protein, we call it Melusin, which is a protein identified as an effector acting downstream of the integrins that are the receptor at the membrane of the cardiomyocytes involved in connecting the extracellular matrix to the contractile cytoskeleton. And the reason why we decided to investigate the role of this protein were dual. One is this protein is cardiac specific, or better, is skeletal muscles and cardiac muscle specific. So it's expressed only in those two tissues. Second of all, it was not known its function at that time when we started to work on this molecule. And so we decided to have a very straightforward approach and performing a knockout of the genes in mice and then performing a gain-of-function model with a transgenic mouse expressing selectively this protein in the heart. And I summarize here what are the results of those studies. Basically, if you knock out the gene, so you have animals that lack the expression of the proteins, no phenotype in basal conditions. However, if you challenge them with the aortic bending, as shown here, what's happening in these mice is that they fail to have a proper hypertrophic response in the beginning and accelerate their transition toward dilation of the left ventricle and reduced contractility compared to the wild type. So in absence of Melusin, the heart undergo dilation and loss of contractility much more quickly compared to the wild type. And the reverse is true, of course, also in the gain of function model, which is the transgenic mouse. Let's see whether this works. Okay. So if you do again the aortic bending model, these are the wild-type mice, and you need to perform aortic bending for a long period of time in order to induce dilation in the wild-type strains here. But if you do the same kind of treatment in the transgenic mice, as you can see here from the echo analysis and from the contractility analysis here, they can keep contractility, well, I mean, at a very good level, while the wild-type fails under these conditions. So basically, Melusin is required to, for the heart to respond properly to the stress conditions, and if you overexpress it, it protects heart from dilation and loss of contractility. We have recently also demonstrated in collaboration with Gerd Hartzenfuss in Göttingen and within a larger European consortium that overexpression of Melusin is also very much protective in the myocardial infarct model. And this is just a mortality curve in mice that have been subjected to permanent ligation of the coronary artery. And as you can see, there is quite a steep drop of mortality in the wild type around day 6 here, which is due mainly to cardiac rupture. But transgenic mice are very much protected compared to the wild type in this crucial events of cardiac rupture in the beginning. And this is illustrated by this simple histological experiment here. But as you've seen here, the number of mice analyzed is quite large, and the protective effect is no doubt there. If we analyze the recovery of the ventricular function in the next 2 weeks after myocardial infarction, the transgenic mice are also performing better. And as you can see here, the dilation of the left ventricle that is, of course, occurring after myocardial infarction is much less in the transgenic mice compared to the wild type. And if we measure the contractile activity again of the left ventricle, it drops after myocardial infarction, but it drops less, of course, in the transgenic mice. So from these kind of analysis, we can conclude pretty safely that overexpression of Melusin is a quite strong protective effect in both the pressure overload model as well as the myocardial infarction model. And what about humans? We have just this analysis that was performed again in collaboration with Vera Zagrasek in Berlin years ago, analyzing the heart explanted from patients that undergo heart transplantation due to heart failure triggered by aortic stenosis. And as you can see here, the level of Melusin, it correlates pretty much with the level of functionality in the left ventricle. So the lower is the ejection fraction, the lower are the Melusin level in the left ventricle. So what about the with the molecular events underneath these protective effects? First of all, I have to say that we have shown some years ago already that Melusin is a protein that belongs to the family of small cochaperones. Small cochaperones are proteins that have a chaperone function themselves, but they interact with HSP90 and most likely help HSP90 in assisting protein folding during signaling. I'm sure that most of you know, but a function of chaperones in general is, of course, to assist protein folding during synthesis, but this is not only their role, their crucial function is also during signaling. And during signaling, protein have to, of course, shift from one tertiary structure phase to another tertiary structure phase, and this transition needs the help of chaperones and cochaperones. And I think that the most striking example is the estrogen receptor, which is usually bound to HSP90, and during estrogen binding, HSP90 is released, and the protein can go to the nucleus and exert its function. But I don't want to give a lesson on that, of course, because I'm sure all of you know. And so we focused on the role of this complex in signaling. And we know from the previous and published study, which I'm not going to summarize here, that Melusin has a big impact in AKT and the ERK signaling. In other words, in absence of Melusin, both signaling pathways are very much impaired in response to the insult, like the mechanical load on the heart. But at the same time, in the overexpressing cardiomyocytes, both signaling pathways are much more effective and more boosted. So the question now is how Melusin can impact on the ERK signaling pathway, for instance. And this is the aspect we have investigated in more detail. So this is the one I'm going to illustrate and summarize to you in the next few slides. So basically, the major start was to show that Melusin indeed itself binds to all the three kinases of the cascade, mainly RAF, Mek, and Erk. So these are immunoprecipitation studies from hearts of transgenic mice in which we precipitated Melusin and then search for a protein that comes associated with Melusin. As you can see here, there is a small amount, but significant and reproducible amount of HSP90. We can find the RAF kinase. We can find the ERK kinase as well as the Mek kinase and the ERK1 and 2 that comes together with the protein. So the co-precipitation is specific, and I have other example of that because, for instance, BRAF, which is also expressed in the heart, is not associated in the complex in this structure. What we have shown here is compare the sham animals, two sham animals, with two animals that have been subjected to aortic bending for only 10 minutes, actually. And if you do that and you analyze I'm just trying to find a pointer that is working better, okay. If you compare the amount of protein associated in the complex after aortic bending compared to the basal conditions, you see a significant increase in Mek as well as HSP90. But we still don't know exactly what that means, but this is a finding. So this is the starting point. The interesting aspect also is that the kinase that comes in the complex are activated upon aortic bending. And this is a kinase assay performed on the co-immunoprecipitation material. And the kinase assay has been devoted to detect ERK activity using this gold standard substrate for ERK. And so these are two samples again. As you can see, there is a nice activation here. If we use the well-known inhibitors of Mek kinase, we abrogate these activations. So basically what we can deduce from this experiment is that upon stimulus, which is in this case aortic bending, very short aortic bending, this is active, and in order to have this activated, you need this protein to be functional, because inhibitors of Mek prevents activation of ERK in the complex. So this is the first part of the finding, but the complex also contains additional components, and mainly I would like to focus on two of them. One is the focal adhesion kinase, which is co-immunoprecipitated with Melusin also, and the amount of this kinase present in basal condition or after aortic bending is not different, so it's a sort of a constitutive complex. And the other important molecule also present in association with Melusin and HSP90 actually is this protein called IQGAP. IQGAP is a 180 kilodalton protein. Its interaction is very strong. This is a Comasi blue stain gels of a co-immunoprecipitated material, and this is the Melusin bands. This is the contaminant Ig present. This is IQGAP that you can really clearly stain with this system. So the interaction is quite stable. Also, in this case, the interaction is constitutive and is not affected by the aortic bending. The amount of molecule associated with Melusin is not increased or decreased by the bending event. The interesting part of IQGAP that probably is not well known to all of you, so this is just to summarize two additional components of the complex. What is the role of FAK and IQGAP? FAK is also responding to the aortic bending, undergoing activations, and this is again a kinase assay that we performed on the immunoprecipitated material, showing that FAK can phosphorylate this substrate molecule that is used in this as a standard substrate for FAK kinase assay. And if we use inhibitors, pharmacological inhibitors of FAK, we abrogate not only, of course, the FAK activation, but also the ERK activity in the complex, implying FAK is an upstream kinase in the complex. So we've shown that this should be active, but also this should be active in the complex in order to have this activated in response to the aortic bending. And what is the role of the IQGAP? IQGAP has been shown by other lab, and then we have reproduced this data to act as a scaffold for the MAP kinase cascade. And a scaffold means that the protein is capable to bind, again, both RAF, Mek, and Erk. And we have shown that this is occurring also in art extract. And we map the binding site using different IQGAP construct to this IQ domain region, which is the central region in the IQGAP proteins. So this is apparently a very complicated complex of molecules. And the role of the IQGAP actually is required for proper activation of the ERK, again, in response to the aortic bending. To prove that and to show that, actually, we used genetically modified mice, again, IQGAP null mice, that were fortunately available from Bahu's lab in New York. And when we crossed these mice to our Melusin transgenic mice, we had a double transgenic in which we lack this component in the complex. If we lack this component in the complex and we do again our assay, so we immunoprecipitate Melusin, we search for activated ERK. Again, we repeated the experiment shown you before, but the activation of ERK is significantly reduced if the complex is lacking IQGAP1, meaning that this scaffold protein is also required to assemble a fully functional signalosome downstream of the integrins in response to this particular kind of stimuli. So coming to the also in vivo situation, is really IQGAP also important in vivo in the heart function? And of course, this slide is to mean that it is, and I'll show you briefly. First of all, IQGAP null mice are perfectly viable, as well as the Melusin null mice I've shown you before, and they have no defects in the basal function of the heart. Even after aortic bending, as shown here, the performance of the heart is not changed in the absence of IQGAP. But if we keep the aortic bending stress for actually a longer time, like 12 weeks here, what we observe is that IQGAP null mice start to undergo dilations and loss of contractility, as shown here, much more dramatically compared to the wild type. Actually, the wild type under this condition do not lose very much in their functions, but the IQGAP nulls have already reached very low level of contractility. So what we conclude from this experiment is that indeed IQGAP is not required for basal function and for initial compensatory hypertrophy, but is required in order to maintain the functional property under chronic stimuli under these conditions. And what is happening in this particular case is that the apoptotic death of cardiomyocyte, which is triggered after 1 week of bending, very dramatically, but almost similarly in the two samples, is much more evident in the IQGAP null after 12 weeks of bending compared to the wild type. So there is a increased susceptibility to cell death in the absence of IQGAP. And interesting enough, what we observe, in spite of the fact that these hearts are doing worse, the level of BNP and ANP are actually lower in the IQGAP null mice after 12 weeks of aortic bending, implying that these molecules, which are known also to be protective in the heart, are actually not properly induced in the absence of IQGAP. So this is not a conventional view of looking at these markers, which are usually used as marker of a failing situation, but actually this is a reverse situation, suggesting that in absence of that proper signaling, or those signaling controlled by IQGAP are required to do activation of these molecules. So what is our conclusion after these kind of analysis and after this complex signalism system? So what is well known is that ERK signaling is activated, as I was mentioning before, by many different stimuli in the heart when subjected to different stress conditions. The angiotensin receptor, for instance, is well known to activate the ERK pathway. Growth factor receptors, like MET signaling, for instance, is well known to activate the ERK pathway, as well as other IGF-1 and so on. But what is different in all these situations is actually the kind of the scaffold molecules that are involved in assembling these pathways and probably driving these effector molecules towards specific targets. So in other words, angiotensin receptor signaling is well known to be a maladaptive signaling in the heart, still is activating also this ERK pathway there. So what we propose here is that we identify by this system a sort of a complex signalosomes which is driven by the chaperone activity of melusin and the help of the scaffold protein IQGAP, acting as a signalosome responding to stress or mechanical stress stimuli downstream of integrins. Part of it is speculation, of course, but it's just as a model, working model here, and is crucial in triggering ERK pathway as well as the AKT, which are under studies at the moment in promoting cardiomyocyte hypertrophy and survival. I haven't shown you the data on the in vitro cells, but we have analyzed that too, and in vivo by triggering the compensatory hypertrophy response. I thank you for this, and I can answer all questions. Thank you.